Everyone, my name is Swetha Ganesan, and today I'm going to be presenting my poster on the development of a standardized protocol for brain slice registration to the Franklin and Paxinos Brain Atlas. You should be able to download a PDF of my poster from the GoReact website, but for the purposes of this presentation, I'll be adapting the content to better fit a PowerPoint format. So before we get started, just a little bit of introduction. Localizing manipulations and features of brain tissue sections is crucial to validate any experimental claims of anatomical or functional connectivity. And neurobiologists commonly achieve this goal by registering tissue slices to reference images in a brain atlas. Numerous labs perform this process of slice registration independently, but this has created a need for a standardized platform to ensure reproducibility within labs. So that brings us to the goal of this project, which is to develop a standardized registration protocol to localize sites of interest in mice brains using the third edition of the Mouse Brain Atlas in stereotaxic coordinates by Franklin and Paxinos. Our protocol was developed in the context of a greater in vivo electrophysiological experiment investigating the connection between the cerebellum and the nucleus accumbens regions in mice brains and how this connection has been mediated by the ventral tegmental area, or the VTA. My co-author, Alexa D'Ambra, who is a graduate student in the lab, has been investigating this connection by first lowering a stimulating electrode into the cerebellum so it can stimulate the deep cerebellar nuclei there, and then lowering a recording electrode into the nucleus accumbens. She then investigates the role of the VTA using viruses that contain DREADs. DREADs are designer receptors exclusively activated by designer drugs, and this virus ideally works by spreading across the VTA and transfecting these VTA cells so that they now express these designer receptors. These receptors can only be activated by a designer drug, which in our case is CNO, and when they are activated by CNO, they shut down the VTA cells. So this allows us to investigate the role of the VTA in mediating the cerebellum and nucleus accumbens connection. My role in this project was to localize the electrode placement. This is important to ensure that we are stimulating and recording from our intended area, and I also helped evaluate the expression of desired proteins in the VTA and validating injection target accuracy of the DREADS virus. Both of these roles are important to ensure that the collected electrophysiological data was actually reliable. So step one of our protocol is to perfuse the animal with PFA. So PFA is paraformaldehyde, and to perfuse it basically means to introduce PFA into the animal's circulatory system. Once we've done that, we want to extract the brain and then store it in a vial of PFA for at least another 24 hours for the post-fixation process. Next, we want to use a mouse brain matrix, shown on the left, and a Vibratome Series 3000 plus tissue sectioning system to section the brain tissue into slices with our desired micron thickness. Once we've collected our slices with the Vibratome, we can put them into a well plate that contains PBS solution. And then we can examine our collected slices under an Olympus SZX16 stereo microscope to identify and then later mount our slices of interest onto microscope slides. We then want to take our microscope slides and look at them under an Olympus VX61VS microscope and use the Olivia VS-ASWFL program to scan and image these slices. So once they're properly imaged, they should look like the slice on the right. Finally, we want to then register reference maps from the Franklin and Paxinos Brain Atlas onto the slice images that we've just collected from our past step to be able to localize the experimental manipulations. An example of a reference map is shown on the right. So standardization is important at all steps of our protocol, but we want to focus on optimizing the slicing and registration steps. For slicing optimization, you want to first start out by positioning the brain in the brain mold such that the blade lines run perpendicular to the mid-sagittal line. Then you want to place your first blade anterior to the brain, and then your second blade in between the cerebrum and the cerebellum. So this not only helps you section off the cerebellum, but also secures the rest of the brain while you slice the remaining cerebrum section with additional blades. Once you have all your desired sections, you can set up the Vibratome 3000 plus system to slice the sections with desired thickness. So for our experiment, we used 150 micrometers, but you can use anything up until 250 micrometers, but beyond that, you might lose whole sections. For registration optimization, you want to take the brain tissue slice images and open it in ImageJ application by Fiji using the Bioformats plugin. You want to then separate the image layers and manipulate the contrast and brightness settings for each layers up until it's the desired level that you want it to. You then want to merge the layers and add a scale bar in the bottom for future reference. And finally, save this image as a .png file. So once it's done, it should look like this. So once you have your saved PNG file, you then want to identify the Brain Atlas reference map that corresponds to the image slice using some reference points. We'll identify a couple reference points in just a bit, but this is the reference map that best correlates to the sliced image. Then we want to open the GIMP application and open the saved PNG file right here in the GIMP application and open the corresponding reference map as an additional layer. We want to then manipulate the size, shape, and overall orientation of the map to best fit the reference points of the map over those of the brain slice. So once the reference map is overlaid like this, you can then begin analyses on the experimental tissue slices. When looking at brain slices in the vicinity of the nucleus accumbens region, there are a couple reference points that we can look at to pinpoint exactly where in the brain we are. One useful one is looking at the distance between the bottom of the lateral ventricle and the ACA, or the anterior end of the anterior commissure. Another good one to look at is the size and shape of the corpus callosum, as well as the shape of the ventral end of the brain slice. So all these reference points change in either size or shape as you move anterior or posterior within the brain, and they are really useful to not only help you identify which reference map best fits or correlates with the slice that you have at hand, but also helps us overlay the map on top of our slice with greater accuracy. In this slice, we can actually tell where the electrode was lowered. So you can tell that the electrode was lowered right here because of the fluorescent dye trace. So the electrode is, in our experiment, was coated with a fluorescent dye. And we can look at this trace to not only tell us where in the brain we are and confirm that we are in the right place in the nucleus accumbens. When looking at VTE slices, a really useful tool to use is TH staining. TH staining stains any dopamine-producing cells, and this is especially useful for our purposes because it can help us delineate the VTA region, which is rich with these dopaminergic neurons. Some really useful reference points that we can look at within the VTA. One good one is the ML, or the medial lemniscus region. So it's actually a bundle of fibers, and therefore it's characterized by an absence of any TH staining. Other good reference points that we can look at are the periaqueductal gray, so the size and shape of the PAG, or the MGN, which is the medial geniculate nucleus, or the dentate gyrostructures. So our registration technique is useful because it allows us to now look at the DREAD expression within the same slice that's already been registered. So the viral DREAD injection is shown in red in the bottom two slices, and we are now able to comment on the viral injection accuracy within the VTA, as well as how it spread and the density of its expression. So this is ultimately useful to evaluate how well we were able to shut down the VTA during the electrophysiological experiment. In the context of electrophysiological experiments investigating functional connectivity, fluorescent tracers and dyes are often used to mark sites of manipulation. With our proposed standardized registration technique, we are now able to localize these sites of interest, and this helps grant us anatomical confirmation of functional connectivity experiments. We aim to reduce subjectivity by prioritizing specific brain regions to focus on while we're matching the slice images to a reference map. And we hope that our proposed protocol has the potential to become an efficacious standard for other labs to follow in the future. Current reference maps from the Franklin and Paxinos Brain Atlas illustrate a quite ideal structure in brain structure that does not necessarily account for individual differences between mice or inconsistencies in brain slicing. In the future, we hope to explore options of customizing reference maps without compromising the established objectivity of our protocol. And a potential future direction we could take this is to produce an algorithm that can manipulate reference maps to better fit our slice images, allowing for more precise registration and increased analytic accuracy. I just want to end my presentation by saying a special thank you to Alexa Diambra, to Yasin and Fioravante, and the entire Fioravante lab for all their support and mentorship throughout this entire project. Thank you all for listening to my presentation.